Welcome to our video abstract of our research paper in brain communications titled Progressive Changes in Synapses and Glial Cells in AppNL-G-F mice, a mouse model of Alzheimer's disease. It's well known that synapse loss correlates with cognitive decline in Alzheimer's disease. However, the mechanisms that contribute to synapse loss remain poorly understood. Amyloid-β plaques and oligomers accumulate around synapses and can directly drive synapse loss in Alzheimer's disease. But in recent years, glial cells have also gained attention as key contributors. However, their role remains unclear, and it's still debated whether their actions are ultimately beneficial or harmful to disease pathogenesis. And so this led us to two main outstanding research questions that we wanted to address, which was firstly, do changes in synapse number occur concomitantly with changes in glial cell populations in Alzheimer's disease? And secondly, how do synaptic and glial cell alterations correspond to the temporal progression of plaque deposition in Alzheimer's? To address these questions, we use the AppNL-G-F knock-in mouse model, which carries the humanized form of APP with 3 mutations leading to amyloid-β overproduction, without overexpressing APP. And examples of characteristic phenotypes in this model includes aggressive amyloidosis, with cortical deposition of plaques as early as 2 months, synapse loss, gliosis, and memory impairment. And so we assessed this model at 3 different time points, 2, 5, and 9 months, examining excitatory synapses, inhibitory synapses, and glial cells, specifically microglia and astrocytes. To do this, we analysed synapses and glia at the cellular level using immunostaining and confocal microscopy, and then further examined glial cells at the molecular level using quantitative PCR. So to briefly explain how we analyse synapses, we quantified synapse number in the stratum radiatum of the CA1 region of the hippocampus, a region where synapse loss is well documented in Alzheimer's disease, and where CA3 axons synapse onto CA1 dendrites. And from 5 months onwards in NL-G-F mice, when hippocampal plaque load is notable, synapses were quantified at different distances from plaques up to 50 microns away. Starting with our results for excitatory synapses, we found no change in synapse number at 2 months. However, at 5 months, we observed a clear decrease up to 10 micrometers away from plaques, and this was exacerbated at 9 months, with synapse loss extending up to 30 micrometers away, demonstrating that excitatory synapses are lost proximal to plaques in NL-G-F mice. In contrast, we found an increase in inhibitory synapse number at 2 months of age in NL-G-F mice. However, no changes at 5 or 9 months across any distance, suggesting that inhibitory synapses are transiently increased prior to substantial plaque deposition in NL-G-F mice. For our analyses of glial cells in the hippocampus, similarly to synapses, we examined both microglia and astrocytes at increasing distances from plaques up to 50 microns away, using concentric circle analysis. For glial cells, I'm only going to highlight cell number, though we assessed coverage and intensity. But starting with our findings from microglia, at 2 months, microglia number was unchanged, but by 5 months, we observed a marked increase in microglia within and immediately surrounding plaques, and this effect became even more pronounced at 9 months. At the molecular level, we found several microglia-related genes change with age in NL-G-F mice. Notably, CD68, a phagocytic microglial marker, and Trem2 was significantly upregulated from 5 months onwards in NL-G-F mice. For astrocytes, similar to microglia, cell number was unchanged at 2 months. However, by 5 months, astrocytes were increased close to plaques within 25 micrometers, and by 9 months this increase extended further out between 25 and 50 micrometers. Interestingly, astrocyte number was decreased within plaques at both 5 and 9 months. Again, at the molecular level, we found several astrocyte-related genes changed with age in NL-G-F mice. Notably a downregulation of synaptogenic markers thrombospondin 1 and 2 at 2 months, and then from 5 months onwards a reduced expression of the glutamate transporter GLIT1, and by 9 months a clear upregulation of GFAP. In conclusion, we define a temporal sequence of changes in synapses, glial cells, and amyloid-β pathology in NL-G-F mice. We found an increase in inhibitory synapse number represents the earliest detectable change in NL-G-F mice at 2 months, alongside changes in some glial cell markers at the molecular level, and this is followed by excitatory synapse loss and cellular changes in microglia and astrocytes, which occur alongside plaque deposition in NL-G-F mice.